In this video tutorial, I am going to discuss about beta oxidation of odd chain fatty acids. In the previous video tutorial, I had discussed about the beta oxidation of even chain fatty acids. Even chain fatty acids are more common in nature than the odd chain fatty acids. Odd chain fatty acids are found only in some plants as well as in some marine organisms. Now, if we talk about the oxidation of odd chain and even chain fatty acid, then there is only a very minor difference. See, to understand what is the difference, let's first consider example even chain beta oxidation. Suppose I consider 8 carbon long even chain fatty acid. So it has 8 carbons, right? 1, 2, 3, 4, 5, 6, 7 and 8 carbons. Of course, this is a carboxyl group and this is the methyl group. Now we know that with each cycle of beta oxidation, 2 carbon units are separated. So first beta oxidation cuts over here, second beta oxidation cuts over here, and third and final beta oxidation cuts over here. So at the end of final beta oxidation, we get two carbon compounds. But in case of odd chain fatty acid, let's make it 9 carbon long. So this is 9th carbon, right? So what will happen at this time? So when final beta oxidation occurs, at that time, instead of this 2 carbon compound, it is 1 2 carbon compound and 1 3 carbon compound gets separated. So this is the unique compound which is coming out of the beta oxidation of odd chain fatty acid. Now what is the name of this 3 carbon compound? So before giving it a name, let's first write down its structure. So this is the methyl carbon, right? So it has a methyl group. The second carbon is a hydrocarbon change carbon. So of course it is CH2. The third carbon is going to be carbon having this oxygen and it is going to attach with the S-CoA. So this compound gets separated and the name of this compound is the propionyl coenzyme A. So as this is the unique compound which is being separated from the odd chain fatty acid, we have to describe its metabolism. So what will happen to this propionyl coenzyme A next? So to describe it, first I will rearrange or rewrite the structure of propionyl coenzyme A. Instead of horizontal, let's write it down in the vertical fashion. So it is 3 carbon. So carbon 1, carbon 2 and carbon 3. On the first carbon there is CH3. On the second carbon there is CH2. And the third carbon there is on one side it has oxygen, other side it has coenzyme A with its sulfur. So this is our propionyl coenzyme A. Now in the first reaction, what happens? Carbon dioxide is being added into this propionyl coenzyme A. So basically as carbon dioxide is being added, we call it as a carboxylation reaction. Now this carbon dioxide is inserted where into this compound? So it is inserted on this second carbon. So how this compound looks like? So here also it is 3 carbons, right? Now the first carbon is unchanged, so CH3. Now on the second carbon, this carbon dioxide is being added. So one hydrogen will be lost, so there will be only CH and then it will be bound with the COO minus. This is our carbon dioxide. And on the third carbon, the structure remains same. So now this compound is called as a methylmalonyl coenzyme A. Methylmalonyl coenzyme A shows a stereoisomers. That means it has 2 varieties, capital D and capital L. By this reaction, this capital D methylmalonyl coenzyme A is formed. Now this reaction requires energy and the energy comes from the hydrolysis of ATP. So one high energy bond of ATP is broken down. So ATP gets converted to ADP and one inorganic phosphate. This reaction requires one very important vitamins and that is biotin. Remember, biotin is required for this reaction. And the enzyme which catalyzes this reaction is the propionyl-CoA carboxylase. So here as you can see, propionyl-CoA is the name of substrate and carboxylase is the process which occurs in this reaction. Now next, what happens to this D-methylmalonyl coenzyme A? So in the next step, D-methylmalonyl coenzyme A undergoes isomerization reaction. That means this D-methylmalonyl coenzyme A gets converted to L-methylmalonyl coenzyme A. So here this carboxyl group will be shifted instead of right, it will be shifted on the left side. So let's rewrite this molecule. So we have first the CH3, second one is the CH. Now instead of this C double O on right, we have C double O on left. And the third carbon is unchanged. So C with C double bond O and S COA. So this is the structure of L-Methylmalonyl coenzyme A. Remember, nothing is added or nothing is removed in this reaction. This is simply isomerization reaction, and this reaction is catalyzed by the enzyme methylmalonyl coenzyme A racemase. Some book also says methylmalonyl coenzyme A, and instead of racemase they use word epimerase. So both names are correct. Now next, what happens to this L-methylmalonyl coenzyme A? This L-methylmalonyl coenzyme A, it further undergo isomerization. Now here, instead of simple isomerization, there will be a molecular rearrangement. That means this carboxyl group now shifts from this second number carbon to the first number carbon. So let's write down the formula, what will happen. So we have 3 carbon, carbon 1, carbon 2 and carbon 3. Now this C double O will be on the first carbon. So this carbon contains C double O minus. And so it has, instead of 3 hydrogen, it will have only 2 hydrogen. Now here on the second carbon, instead of 1 hydrogen, we have 2 hydrogen. The third carbon remains unchanged. So C double bond O and then coenzyme A. The name of this compound is Succinyl-coenzyme A. This reaction is catalyzed by the enzyme methylmalonyl coenzyme A mutase. And this enzyme needs vitamin B12, the coenzyme of vitamin B12, as a cofactor. And the coenzyme form of vitamin B12 which is required over here is the deoxyadenosyl cobalamin. Now what happens to this succinyl coenzyme A? Succinyl coenzyme A undergoes TCA cycle to help in the oxidation of acetyl coenzyme A. Succinyl coenzyme A is also a gluconeogenic substance. That means succinyl coenzyme A can undergo multi-step process and can give rise to glucose. Now this is very surprising. Remember, it is generally stated that fatty acids can never generate glucose. But this is the one exception, that this succinyl coenzyme A can generate glucose. So this you have to remember in the form of exception. And this pathway by which glucose is generated, it is called as a gluconeogenesis process. So basically, odd chain fatty acid can give rise to glucose. Now let us discuss the applied aspect or clinical importance of this odd chain fatty acids beta oxidation. See, there are two conditions. One is vitamin B12 deficiency, and the second is the mutation or deficiency of this methylmalonyl coenzyme A mutase. So deficiency of enzyme methylmalonyl coenzyme A. Now in both the condition, what will be happen? This reaction, this step will be blocked. So obviously there is increased concentration of methylmalonyl coenzyme A. So this both condition has the same effect, and that is increased concentration of methylmalonyl coenzyme A. And when this methylmalonyl coenzyme A is accumulated, it spontaneously gets converted to methylmalonic acid. There is a removal of this coenzyme A with its SH group, right? So obviously if this methylmalonyl coenzyme A is accumulated, there will be increased amount of methylmalonic acid. Now this first increase in the blood, and when anything increase in the concentration in blood, we generally use suffix amia. So this condition in which our blood contains more amount of methylmalonic acid, we call it as a methylmalonic acidemia. And if it is increase in the blood, then eventually it leads to increased excretion in urine also. And when something increase in urine, we use suffix urea. So this condition is known as a methylmalonic aciduria. Now see, this is important, that is methylmalonic acidemia. This is methylmalonic acid is acidic, so it leads to decrease in the blood pH, and this condition is called as a metabolic acidosis. This is very dangerous condition because we know that all the enzymes on our body, they are very, very sensitive to the pH, to the acidity or alkalinity. And if pH of blood is shifted, then most of the enzyme in our body stops working or not properly functioning. So this is very serious condition. This also leads to CNS damage, nervous system gets damaged, and in the small children, it leads to growth retardation. So there is a growth retardation. And if this is not treated, then it is going to be a fatal in early life. So that's all about the beta oxidation of odd chain fatty acids. I hope that all the concepts are clear. If you still have any query or confusion, please write it down in the comment section below. Thank you.